Alrighty, folks, grab your lab coats. We're gonna dive into the fascinating world of genes. Today, our star is the hemoglobin gene, responsible for oxygen-carrying protein found in red blood cells. But before we can get to the making of this vital protein, we need to follow a journey of hemoglobin gene through the DNA replication, transcription, and translation. Think of this as a cross-country road trip, only instead of stopping for snacks, we're building proteins. Let's hit the road. Okay. So our adventure starts on chromosome 11, where our hemoglobin gene is located. This gene holds the instruction for making hemoglobin proteins. But first, before anything happens, this gene is all wrapped up in a tightly coiled DNA. And as we've learned before in our previous studies, we can't really get to work if the DNA is in a knot, right? So this is where the helicase enzyme steps in. It's like a zipper puller, and it kind of unwinds the double helix of the DNA and creates a replication bubble, giving us access to the gene we need. And this is our cue to start copying. Now that the DNA is open for business, we need to copy the gene. This is where DNA replication comes in. Think of this as making a backup of a super important file because this gene will be passed on later. Enter DNA polymerase. This enzyme moves along the open DNA strand and adds complementary nucleotides to a new strand. But wait, there's a twist. There are two strands to copy. The leading strand is copied smoothly and continuously, while the lagging strand is copied in chunks thanks to the Ozaki fragments. It's like one side of the highway is under construction, but we're still kind of getting things done, you know? Eventually, the two strands are fully copied, and now we have two identical copies of DNA. They're ready for action! So then, with our DNA copied, it's time to create the messenger. mRNA is like the notes you take during a lecture, only instead of scribing it on a paper, we are using RNA to write down the instruction for protein production. This process is called transcription. Inside the nucleus, the enzyme RNA polymerase gets to work. It attaches to a specific spot on the DNA called the promoter region, kind of like finding the perfect starting point. Then it reads the DNA template strand and builds a complementary strand of RNA. This is called the elongation phase. Just like building a train track, RNA polymerase adds nucleotides to the growing mRNA strand, and eventually it reads the terminator sequence, which is basically the finish line. The mRNA is released, the DNA is coiled back up, and it's ready for its next round. But before our mRNA leaves the nucleus and heads to the ribosomes, it still needs some finishing touches. Think of it like getting ready for a big job interview. I have one in a few days. First, a 5 cap is added to the beginning of the mRNA to protect it and help find its ribosome. Then, a poly A tail is added to the other end. It's kind of like a VIP pass to get into the ribosome club. Finally, we do some cleanup, which is splicing. The introns, the known coding parts, are cut off, and the exons, the coding parts, are left to make the final perfect mRNA transcription. This mRNA is now ready to leave the nucleus and make its way to the ribosome in the cytoplasm. I hope you remember your cell parts. It's sending the mRNA to a protein building factory. Okay, now we're here at translation. Welcome to the protein factory. Here is where the magic of translation happens. The ribosome, which is like the assembly line of the cell, grabs onto the mRNA and starts reading it. The ribosome has two key areas. The site A, where tRNAs bring in amino acids, and the P site, where the growing protein chain is held. As the ribosomes reach each codon, which is a set of 3 mRNA bases, a tRNA matching with the amino acid enters the site A, or A site. The ribosome then shifts and moves along the mRNA from 5 to 3 direction, and the mRNA moves to P site, where the amino acid it carries is added to the growing chain. This continues in what we call an elongation process, with the protein chain getting longer and longer. Once the ribosome hits a stop codon, translation stops and the protein is released, ready for its next steps. But wait, our protein isn't just ready to work yet, it has to fold into its final shape. This is where the protein undergoes post-translational modifications, like adding sugar groups or phosphate groups to make it more functional. Think of this as making sure a piece of furniture has all its screws tightened or etc. Without these modifications, the proteins wouldn't work properly. Some proteins, like hemoglobin here, even undergo changes in their structure to help them perform their job more effectively. It's like fitting the last pieces of a puzzle together to reveal the final picture. Now that our hemoglobin is fully folded and ready to go, it has a very, very important job to do: oxygen transport. Hemoglobin binds to oxygen in the lung and then it's off to the bloodstream to deliver the oxygen to the tissues that need it. Without hemoglobin, we wouldn't be able to breathe, or at least not for long. And to sum it all up, all of these steps sounds like a lot of things happening, but we have to think about how it constantly goes on in our cells. Hemoglobin's job is essential. Without it, oxygen wouldn't be able to get to our organs and tissues, and our body would struggle to function. Maybe not function at all. So the next time you take a breath, remember it's a result of an intricate, well orchestrated series of processes, starting with a single gene on chromosome 11. Pretty cool, right? That's all for today. Bye!